drug formulations from plants, individual as well as formulated drugs. And these drugs, we wanted to test them against human beings. We've done it after a brief toxicology profile done with animal models, as well as again came back to clinical studies straight away, because that was a period where no clinical studies were necessary or mandatory for herbal drug. 2008 only it was all formulated to have this herbal drug also. During that period of time, we have conducted more than 15 clinical studies. But we have conducted 15 clinical studies, though we have done that. We have done it in reputed hospitals like NIMHANS, St. John's Hospitals in Bangalore, Government Thoracic Hospital at Tambaram Sanatorium. We did it in KG Hospital in Coimbatore. We did it in several Lucknow hospitals, so many different AIMS also. So we have done a lot of studies in the clinical studies, proper clinical studies in comparison with an allopathic drug wherever is possible, unless otherwise we do placebo controlled clinical study. So this was with the brief background. I thought I can straight away go into the purpose of my talk. Yeah. See, normal drug discovery process, as Professor Kinney has earlier mentioned, it comes to $60 million. Yes, truly speaking, if you calculate the above, total 60 million, so approximately $60 million, which is close to about 500 crore rupees for a drug to be developed in a pharma company. Hence, they have to invest that much of amount of money. The charges when it comes as a drug is going to be also higher. And you see the failure rates in the 60% of failure rates starting from, and there is a final phase we have got only 10% of failure rates. So this is how it happens, and you see from pharma companies. This is to understand how a pharma company is approaching a drug. See, high throughput screenings is first phase, and second phase is preclinical phase, and third phase is human phase, where phase 1, phase 2, phase 3 and phase 4 studies are being happen. So all takes about, if you calculate 12, 13 years, it will take for any drug to come into the market. So the failure rates are higher, the amount is high, and the time duration is high, hence the price is also high. Yeah, latest, I just wanted to give you another one brief information on latest trends in pharma for companies. What are the latest trends in that? They want global and decentralized studies to be the standard, because when they are doing actually a clinical study in abroad or in India, they don't accept. One person, US, if you did the study, India doesn't take out this one, the study details at all. They want the study to be conducted in India. So the first one foremost they wanted is global decentralized studies have to be the standards. That is what they wanted to make it. And radiomics, they are doing it based on the image studies, based on the image results, they are developing the drugs. So, and then natural language processing, which is nothing but using artificial intelligence. Most of the pharma companies are using artificial intelligence to decipher a drug. And priority shifting, yes, today they have a priority of some drugs, something had to be done, and tomorrow the priority may shift. Because for example, when it comes to this coronavirus which came into that, all priorities have been shifted to coronavirus. So, and digital therapeutics, you all might be known about it because you would have heard this information, because this is what is going to be. These are all the topics which if you get familiarized and if you get trained, get proper knowledge, if you gain that, then it would be easier for getting into pharma companies now. It is very, very easy to, if you do these things properly, study properly, make things properly, and get yourself trained in that. And digital inventory management is one of the area, and another personalized treatment medicine. This is where now people started looking into personalized treatment medicine, because each person is having differently. Because this how it happens, because well, we have been, see our genome, if you can notice the genome, actually the expression of genome, epigenetics we call it as, the epigenetics majorly rule your health or wealth, health or your character or anything. It is not the DNA alone. DNA only contributes about 25%, whereas epigenetics contribute about 75%. So it is that is the reason why personalized treatment medicines are absolutely necessary, and Ayurvedic science is doing that, because they don't treat the same person same time. That is, same medicine cannot be given to same person. It should be something different should be kept into. So that we have to take it into consideration, and yes, precision dosage and monitoring of specialty drugs, that is also their area, and expecting the unknown also there. Yeah, in this one, if you take the case of plant-based medicine, reverse pharmacology works very well, because that is what we have been doing it for the last, during the phase of 1995 to 2010. What we did was, we deciphered based on the ancient literatures and bring back medicine back to our own proprietary formula, and we do testing with animal models and found out to be non-toxic. You should have the maximum, at the time it was 500 milligram per kilogram. If it is able to tolerate, the drug is, the animal is going to tolerate that much amount of drug, we call that as it is a non-toxic drug, and then we can straight away go to the clinical study. Straight away we go to the clinical study part of it, and then we found that is efficacious. Then we see the phytochemistry aspect of it and see which compound is doing what. This is the reverse pharmacological approach. This is what the different steps which has been involved, which has been given here. If any of you are interested, you can get it from that laptop. So we call it as starting from identifying the disease. This is the most important thing, because identifying this disease which has got a proof, which is already having no side effects, is already having efficacious than anything, and the cost is less. We don't touch that disease at all for our research. We take it in a way that where allopathy is failing, or allopathy is not able to make up the issue, we take that into consideration, like side effects or anything. More side effects are there in certain drugs, then we take that disease or disorders that can be used, and we find how all these how we develop the formulations, and chronic toxicity, dosage correction. These are all things which happens nowadays. CTRA has come in, that is central, this one I forgot, Clinical Trial Registry of India. Clinical Trial Registry of India, institutional robots has come into the picture for making that with the registration, and then protocol. We also decipher protocol, because based on when we take up the allopathy drug in our arm, one of the arm of our study in a clinical trial, what we do is actually we take the same pharma route, that is the protocol is designed as a pharma route, and then we efficacy of our formulation. This is what we do that. And finally, patent, market, and then phytochemistry comes into picture. So it is called as reverse pharmacology. Okay, why do you have to go for plant? This is the first question which I wanted to ask yourself, and then there is a need for it. Why means that what is the need of a plant drug? Plant drug is complex structural diversity it has got. Because any inspiration, you can see here bottom, 60% of all medicines are either from natural or secondary metabolites only. No, without plant, a drug cannot be made. In earlier days, we had aspirin, digoxin, and morphine, Taxol, quinine, any name. Even today, those drugs are there, alive and doing well. And all these drugs are coming from nothing but plants. And it has got a very unique compounds, because approximately around 10,000 compounds are minimum. 1 lakh compounds are present in each plant. All are different from, some are common, like beta cytosol, or some compounds are common between that. most of them are specific to the plant. If at a cucurbitaceae family, then cucurbitaceae A, B, C, D, E, F, so many cucurbitaceae in different concentration in different plants. That is why the treatment has to be, the plant selection of the plant has to be properly done. And it is also from undiscovered resources. That is, if you take the case of plant, you know how many plants are there? Answer this question. Approximately, judge in the world. I am talking about species. You don't want to guess at all. It's about 5 lakh, okay? 5 lakh plants are there. You know how many plants are being used as drug? Plant drugs today? 1,500. That's it. So you don't have, you have lot of scrutinized plants where it could be also useful. So we don't, our next difference is one plant with one combination do something, with one plant with another combination will do something differently. That has to be also noticed in that. So one is novel compounds or novel combinations, new compounds. If anybody wanted to look for new compound, they have to look for nature only. They cannot go anywhere else. Nature means mainly plant only. Because in animals, you do, still animals have got some medicinal value, or marine plants, even that is coming under plant kingdom only. Animals, there are only very few, this one, like vitamins, minerals, some proteins are given, but not like for diseases or problems. Curing, you cannot depend on animal things also. How a plant becomes a drug? This question should come to our mind. When do you consider a plant as a drug, or how it can be? Efficacy has to be proven through literatures and practices. Why I put practices also? Published informations nowadays we are getting it. Earlier days there were no publishment, only by practice people have written notes and things like that. Even folk medicines, if you take the case of Ayurvedic medicine or Siddha medicine or homeopathy, whatever it is, any form of medicine you can take that, and that has to be, the efficacy has to be proven of individual plant or multiple plants. And symptoms it can control, what are external or internal. And toxic studies already done or not to be done, or not done, then we have to do it ourselves. If the formulation is a formulated one or a single, if it is available, availability is more important in this, because though if you take the case of a plant which is going to cure you a cancer, if you talk about cancer and if you say that a plant is going to cure the cancer, but you can't, the plant should not be available only in Alps mountain or Himalaya, because it can grow only there, means there is no use, because who is going to cultivate there? Who is going to trade that? The availability has to be plenty. It should be cheaper for the public to get it. So, or it should be cultivable. It should be tradable or it should be cultivable. And we should also finalize standardization of assays. What assay we have to find out to standardize that, and how efficaciously we have to prove that combination or a single drug is going to be efficacious. And assay guided fractionation or extraction had to be done, because the bulk people cannot take it. In the case of 1 kg of a plant material, you will get around 100 grams. So that is fine. But 1 kg, what you are going to take is per day about 1 gram or 1.5. That should equal to at least 150 grams of the equivalent of plant material, because 10% of some compounds have at 6%, some compounds have got over 18%. So averagely 10%, if we take the calculation, that bulk has to be reduced in the form of when you make it in the form of a syrup, or when you make it in the form of a capsule. So that has to be done. Assay guided, when you are doing it, you should not lose the activity of it. Efficacy should not be last. So, we have to identify assay-gated fractionation, and we also should have a fingerprinting profiling, which these are all things which we do it regularly in our place. This is what phytobiochemistry fingerprint profiling or the determination of AI to be done with chromatographical instruments. The fingerprint profiling is to ensure every time when you are taking a batch production, you ensure that the profiling is matches to the existing earlier one. If it is not matching, then you should put up the plant material in such a way that it is matching. The concentration, for example, concentration of the plant in a particular season is different of a compound. In a particular season, it is different. For example, we have found out in a plant, Kalonji, we call it as in common name, Bryophyllum pinnoitum. It's an, I think you all know Tamil. If you had earlier days, ஒரு சின்ன ஒரு இலையை தூக்கி ஒரு பிளான்ட் ஒரு இதுக்குள்ள நாங்கள்லாம் வச்சுருக்கோம் ஒரு குள்ள வச்சுக்கிட்டா ஸ்டார்ட் புட்டிங் குட்டி போடுவோம் கட்டி போட்டால் குட்டி போடும் அப்படின்னு சொல்லுவோம் அந்த பிளான்ட் இஸ் அ 4 carbon plant. That plant is given actually stone, it is quite useful plant, and that plant has got in the morning, in the afternoon, in the night, different profile of chemical profile we could be able to see it through HPTLC. Concentration wise it is different. So we should ensure that the concentration is properly maintained, which time you should take collection of the plant. Ayurveda specifically insists that such and such a date, such and such a time, certain plants to be collected. This is for no reason they have done it. Without they may be not knowing the exact science behind it, but they have written that when you go through this process of identifying plants, the plant will be shown into your eye only when you go in the morning, when you go in the afternoon, when you go in the night. That's what they mention it. How do they mention it? I don't know how, but I am able to see that. And acute chronic toxicity in animals, yes, we do that. And then we talk about clinical phase 1 to phase 4 and registration and marketing. That's all. We don't require any other thing during a period of time. Now CTRA, the clinical studies have to be registered. Other than that, there is nothing much has been added into new. Yeah, see, I am not going to touch upon the diseases which I already listed out about more than 15 diseases I worked on. I am not going to touch upon. I am going to talk about the latest one which we have formulated last couple of years, which we have been working on, is inflammatory mechanism in plants, human being. How the inflammatory mechanism can be subsided. Because inflammation is one of the reaction by our own body when anything comes from external or internal change. When there is some changes of routine is being changed due to the food that you are taking in or the external agent which are coming into your body. Immediate reaction of the immune system is the inflammation. It immediately starts seeing that extra which is happening there and it goes into it. That inflammatory diseases starting from cancer, the lowest it can go into is asthma or allergy. Starting from allergy to cancer, all are called inflammatory reactions of the body. And cardiovascular disease, everything. So in lung diseases, you have COPD something. In LAM, I will talk about LAM also because one of the patients which we have done it with LAM. And it is in the lung diseases, they have quite a long list of diseases also happening, which currently a patient is being treated with sarcoidosis also. Not with lung sarcoidosis, it was a liver sarcoidosis patient we have been able to do it. And obesity and neuromuscular disorders has also been connected with inflammation only. You see here the picture, the inflammatory genes and differently expressed genes and other genes, how difficult to control this inflammatory mechanism is. This is so tight, so much of diversified and tight. It happens all over the body. For example, if you have noticed in COVID-19 infection, we had you would have heard about a word called cytokine storm. Cytokine storm is nothing but when you take it in, when an infection of the virus came in, our body, it recognizes not just an infection, it recognizes it's a super infection. What happens is, it starts multiple inflammatory genes have been started putting it, and inflammatory genes subsequently targets other inflammatory genes, and then all sorts of network of inflammation happens. That is how we lost SPB. SPB died due to cytokine storm, which happened in all over the body to him, which he could not do it, because the steroid which has been given during that period of time killed so many people, including my father, because he was quite good. I have given him the drug, and his SpO2 level and everything was good. But when he had went to the doctor and got prescribed with an antibiotic called azithromycin, when he took azithromycin, dysentery came into, where he was in Chennai. I was not near him, so I could not travel to him there. And then when he got hold of it, my sister, who didn't have any male member in her family, so what happened is, when he was putting dysentery, she could not handle him. She put him in the hospital, and hospital, without our knowledge, they started giving, and because after 9 days only the steroid should have been given, but on 5th day he has given, 9th day he collapsed and died. Though technically that day, he was also speaking to me at 8:30 night, at around 12 o'clock he passed away. So why I am saying this is that inflammatory mechanism cannot be curtailed by allopathy drug. There is no proper anti-inflammatory drug in the market. You have a painkiller, which is Brufen or something like that, you have, but those are all non-steroidal anti-inflammatory drug, we call it as NSAID, but still it is very, very limited. It doesn't have a broad anti-inflammatory reaction. So where we have to depend totally on plant system. How do you identify inflammation? There are several markers to identify: blood test, ESR, CRP, plasma viscosity, neutrophil to lymphocyte ratio, others small time this one. In this, if you take the case of ESR, erythrocyte sedimentation rate, this is a general inflammation. That is, any inflammation happens in your body, any inflammation, it will increase. General inflammation, not specific to anything else. Whereas CRP is C-reactive protein. When you got infection, then the CRP level happens in the body, then CRP will increase. If CRP increases, that means C-reactive protein. Then if ESR and CRP both increases, that means inflammation due to infection. Whereas procalcitonin, if it is increased along with the CRP and the ESR, procalcitonin also increased, that means you are infected with the bacteria which is causing a sepsis. That is where the sepsis happened in the body of SPB. One more thing is also IL-6, interleukin 6 we call it. Interleukin 6 is the major culprit in this, we can say that it's a central portion of an inflammatory gene. It can, I think more than hundreds and thousands of genes it can trigger. IL-6 is the most culprit in COVID-19. COVID-19 people are having IL-6 higher. When we treated a COVID-19 patient, we asked them to check for IL-6 with them, and we found the IL-6 was around 150, which normally should have been around 40. 250 it was showing, and after her drug, on 5th day she came down, that lady came down to around 64 or something. So this is called, this IL-6 family, it triggers so many, this family contains so many genes that triggers autoimmunity, that triggers allergy, that triggers Th17 dependent autoimmunity and immunosuppression. These are so many different things which IL-6 can do. We have to control IL-6. IL-6 should have been there. It's from normal 0 to 43. It is the normal range. If it is a normal thing, then IL-6 functions beautifully for our protection. But when it increases, it is the problem which we are facing. And I have recently treated 2 patients, one with 43 years of eczema. First we will see this, and then we go to erythrodermic psoriasis. Both the patients have got IgE level, because this person, both of them got, this person has got allergic, because this was 43 years of eczema he has got. His hands were like this, and after our 12 months of treatment, because since 43 years of eczema he had, I had to treat him for 12 months, plainly internal only, no external has been given to him. It takes 12 years to get into correcting. But still, when I checked the IgE level of that person, initially it was, what it should have been is 140 units it should have been, and what it was 50,000. That patient had 50,000, very much, which was very high. So I don't know how I could be able to do it, but I was consistently given that, and after 1 year, he got it down to 16,000. But that is the photograph which has been showing. After the 16,000, you see here, after that it is showing. But still he has not corrected. He has not, his IgE level has not come down. I have asked him to take the drug. He has taken the drug, but he didn't take the test. Because IgE test is around 3,000 rupees per test charge. He doesn't want to take it. That's the reason. So that person actually from Bangkok, he worked in cotton mills. The cotton fibers was the major problem. He continued to take my medicine. He was also continued to be there in the mill, working in the mill. That was the problem. He was continuously exposed to that. So when he started, even if after a while you are taking drug for inflammation, you should avoid in taking that inflammation causing material. Either it can be a cotton fiber or it can be a food which is allergic to you. You should avoid that and then take drugs from plants. That should give you a better solace. Like we were able to do it, and erythrodermic psoriasis just in this year, in the beginning of this year, around June 2024, which I mentioned it there. You see here, June next to, I think, showing. Okay. Yeah, it's not showing. Okay, first. Yeah, actually, if you say first photograph is the June 2000, and next one is July, September, and this is November, last month. And yesterday he sent me the picture. It was further reduced to normal in hand and everything. So it has shown nearly the markers are seen. See, psoriasis, there is no treatment for psoriasis. As you are, I don't know whether you are aware or not. It is only given, the treatment for allopathy is given only as steroid. Right? Steroid. Till you take the steroid, it will be slowing. It will not be, and when you stop steroid, it will again flare up. So we were able to clear up within 4 months with that, and I don't know. Still, his IgE level is more than 400. It came down. It came down to 63 because it was less than the normal. It has come, but his CRP level has not come down. His CRP, I had to bring it down. That inflammation marker, ESR and CRP is still higher, so I want to bring it down. So I asked him whether last time CRP, when he had higher CRP, because it was coming down only, but it was last month it went up. I asked him what is it, because he is suffering from cold. When he is suffering from cold, CRP also some infection and it goes increased. This I have already spoken, and Dr. Victor would have seen, heard in the last my talk also. There's a girl called 25-year-old girl in Coimbatore. She was admitted in the hospital nearby, and she was having, I just explain what is that drug is, what is that problem is. Lymphangioliomyomatosis is commonly called as LAM. It affects 1 in 4 lakh adult females. It's a rare, progressive and systemic disease. You don't have any drug for treatment. The only drug which is given is called sirolimus, which is anti-transplantation rejection drug. That is the only drug which is being given to people, but it doesn't help to anybody, but it only prolongs. this one, what it does is, there is a formation of cyst all over the lungs. A cyst which forms all over the lungs to her body, and in advanced stage it has to be lung transplantation is the only solution. That girl, 20-year-old girl, was actually had pulmonary tuberculosis in January 2020, and during that period of time, she had when detected in June, she had pneumothorax. That is, one of the cysts gets broken. The air goes away through the lungs, then pressurized back the lungs. This is called collapsing the lung. Pneumothorax name is that. That she had a problem, and at that time, that is around July, we have given her the herbal drug, and in the right portion you can see in another 1 year time, the patient 100% got recovered. She was suggested, that girl was suggested to have lung transplantation, asked the farmer of that Gopichettipalayam person who is his father, asked him to register for KMCH for transplantation, which is going to cost them 150 lakhs. 30 lakh has already been spent for the 3 months they have already, because she used to be, she had her thoracotomy here, and then for external oxygen supply. So when I met her, she was 25 kg, and when they have given all this information, I told them that I have got a drug which can function for lung. I did not know whether this drug is going to work for lymphangiometastasis. But that was the time I told them that lung improvement I can give. Apart from that, few more extracts of plants which are safe I can also give, because they have no choice, because ulnar service they have to go for a lung transplantation. They said last attempt we will try to do that. And what happened, last January I met her in her house. Her weight was 64 kg. She is doing very well. And she was taking sirolimus. I asked her to stop it. They were very hesitant because they are from farming community. They said it is very difficult for them to accept this drug will not work, suggested by a doctor. Because this doctor has already suggested them that she will get back this problem again if she stops sirolimus. Then that girl I told her that I have stopped my drug. She was given only 1.5 years, and I asked her to stop it. She didn't want to stop it, but still I stopped her. I stopped her sending medicine. So finally she stopped it. And then she was only taking sirolimus. And then I asked her to stop sirolimus, but she didn't. What happened is her periods were not coming. So they wanted her to get married because she was 27 that time. So they cannot do it because her periods were not, they cannot tell to the people that it is her problem. I suggested her sirolimus. When I searched in the internet, I found that it is known to stop ovulation. It is the side effect of sirolimus. So I suggested them to drop it. So finally in the last January, I asked them to stop it. They stopped it. She said she is having enough, about a week dose or something. After that, she said she stopped it. After a month, she got her periods. Now we are looking for a good bridegroom for her. So yeah, it's good. So I think it was all resolved, 100% resolved. During in 2021 itself, she had 54 kg, starting from 25 kg. The X-ray, which was taken 1 year, this was in 2020. Left one is, and right one is the clear lungs or chest area. You can see the clarity of it. This is the problem. This center is the major issue where she has got a lot of cysts around. You see the clarity here. So here, so this is how it happens. Yeah, another major inflammatory work which we have done it in rheumatoid arthritis, which is known for inflammation in joints, and this inflammation will be painful, swelling and stiffness will be there. So generally more years, there is more than 60 years, people are, the disability is caused by this particular problem. It is, according to allopathy, it is caused by autoimmune. present day treatment, nothing but NSAID as a good pain relieving NSAIDs and disease modifying anti-rheumatoid drugs like methotrexate. These are all monoclonal antibodies, and their toxicity is being listed out here itself. And finally, joint replacement is the only solution people suggested. And our trial contained the methotrexate DMRD in our trial, and placebo and polyherbal have been used. We have also given additional drugs if the person is having some pain again. We used to give them the NSAID, that is ibuprofen type of painkiller, we used to give it. So this trial was conducted in KG hospital, where we have done, you can see the red line which is coming, green line, which is, red line is the placebo, blue line is the allopathy, and green line is the drug, which we, the first one is pain intensity, second one is morning stiffness, third one is RA factor, and the bottom figure is tender joints, swollen joints, general health. In the middle is the herbal drug. That shows the clearly the difference between the this one previous, that is before treatment and after treatment, how much the swollen joints in counts and general health in percentage score has been improved. You can see this. We found this drug, we were doing it, because I don't want to go into much details of it, so it will get boring for you. So I just wanted to give the results to you. There is, we found the last one, you can see that improvement in 70%, 20% improvement is the one methotrexate has been put in for, allopathy drug. We got in some patients 70% ACR, that is Association of American Council of Rheumatologists have given this percentage criteria. Based on that, we have calculated and found our drug comes into 70% category in many of the patients which we have studied. And not only that, we have to also put your head where this can be marketed and how the market potential is. The drug, even though you make it, if the market potential of the drug is very low, there is no need for you to produce that kind of a drug or do research of the drug. You have to ensure that you have a proper marketability of your drug, and you also should know who are your competitors. When you make a drug like that, you have to find out these things. What uniqueness in our product was, we did scientific validation, especially from fresh plants, because arthritis, when we make the oil formula, we made oil formula as well as internal formula. When we made oil formula, internal formula, we ensure that oil formula is made from fresh plants. Yeah, otherwise, in other places, you can see oils are made from dry plants. Fresh plants may have a different structural compounds and different efficacious this one rather than dried plants. And we did it through pure Ayurvedic procedure, and all of them are natural. Yes, this inflammation part I am completing it. Another 10 more minutes, though, should be fine. Yeah, I think this one, another one or two maybe. I have done this one work on antibiotic discovery also. This was done with a patient also. We have done it with a patient who was in ICU, who couldn't be relieved of an infection, came from ventilator. It's called Acinetobacter baumannii, one of my relation. We have given the they have given the antibiotic was not functioning. It was only able to maintain that person, but the temperature was around 105 degrees centigrade, and that person got relieved of the plant compound which has been purified by me. During that period I was working on asthma, so respiratory illness, where I found out a plant which is good. So we extracted the compound, that purified it to 95%, did NMR studies and everything, and then gave 100 micrograms, 1/10 of a milligram to the patient along with antibiotic, and he has got relieved out of the fever, which was hovering around 105 degrees. Kept it in a separate chamber he was. So that made me to look into the antibiotic discovery pattern. At that time I found around 30 years there was no new class of antibiotics. Existing antibiotics had already been resistant to, and there was no new class of antibiotics. We are giving new antibiotics, but it's not, they are not new. What happens is they are nothing but the existing antibiotics. For example, if you have penicillin, you don't have a penicillin, you have amoxicillin. It's the same only. The target is the same. But you don't, only the efficacious is little more efficacious, little less efficacious, something like that. So we need to work on certain plants which can promote or which can do specific function of to potentiate antibiotic or to do something that antibiotic works. That is how our target was. So after 1993, there was no new class of drugs identified so far in this antibiotic discovery. You also should know that United Nations have given a list, have given a report saying that this particular problem of multi-drug resistance or antimicrobial resistance is going to kill the human being in 2050, about 1 crore every year. In India, EDC has already told 20 lakh per year is going to die from 2050 onwards. We should ensure that we should have a proper antimicrobial discovery. This is the patient where we found very simple to identify. You don't need a complicated laboratory to do the job. You just need, you see the plate. Petty plate itself, you can identify the compound which is going to be our plant extract or whatever it is going to be useful or not. By putting an antibiotic disc, you find there is no zone in the left where you, along with the particular compound, you get a zone. That's it. First phase is over. This is all you have to do. Screening multiples of plant and extracts and you start purifying the compound and then you can start doing it, whether that compound is working or not. So these are all the different bacteria which World Health Organization has listed out, about 5 bacteria which are potential. They either have a efflux pump in them, because the antibiotic which goes in, the bacteria effluxes is out. It is an energy dependent process by the bacteria which is doing it. There are 6 different types of pumps which are available in the bacterial system. And but if you are able to stop that effluxing this one, if any compound is stopping the efflux of the bacteria, that will kill, that antibiotic itself will kill. The antibiotic which was not functioning earlier, I want to make it function. That is how I think that person was saved because of that. So the efflux pump inhibitors, antibiotic potentiators can be done. Very easily it can be done through disc diffusion. Efficacy can be analyzed through MIC, minimum inhibitory concentration, and efflux pump inhibitor, say, using ethylene bromide. Ethylene bromide and that is efflux pump function, both are having the same protein only. The same protein effluxes the ethylene bromide also. Ethylene bromide can be very easily measurable using fluorescence spectrophotometer. So that is how we can easily analyze that. So we found certain compounds which are useful. I have already filed the patent for the compound as well as the formula also, because in India only the formulation can be patented, not the compound. The compound can be patented, plant compound can be patented in abroad, but not in India. So we filed the formulation. And approximately I came to the last part of my talk, gut microbes. This is also one of the major things which I have currently been working on. I am yet to develop a formulation on that. I just wanted to put you in this. The left figure saying that, and the red color one and the green color one and the rose colored one. See what happens when some inputs are given, that is like short chain fatty acids. What is the gut microbes in the large intestine does the job, how it talks with the immune system of our body. Everything has already been established, and what food you have to take to maintain the gut microbes, and how certain diseases can be avoided, certain diseases can be controlled. Like for example, I have given you the list. Diabetes is characterized by firmicutes. By taking milk, you can actually control diabetes. And by taking apple and berry, with the seafood and meat, you can control asthma. By using propanobacterium, you can take B12. B12 is very, very low, it means you don't have to worry. You just have to take cheese, milk or milk products, that will do good for you. Because you have got every B12, it is having a lot of B12s in that, so very easily you can have it. And generally, there are a lot of publications saying that how a plant-based people, that is food-taking people, have got a different profile of bacteria, a different profile of bacteria for the non-veg taking. North Indian have got different, and South Indian have got different, and Kerala it has got different for this one, and we have different here. See, these things had to be taken. These anaerobic microbes which are present there, they convert certain amino acids like tryptophan. They convert these amino acids to GABA, serotonin, so many good hormones they are converting into. So 80% of our hormone needs are being satisfied by the bacteria which is present in large intestine. We need to be very, very careful when we choose a food and things like that, so that our gut microbes have done very good one. You have been doing very well. And then plant foods, that is, what is the fermented foods like yogurt, kefir, kombucha, so many things are there. And yeah, these are all areas where you can think about, you can get expertised into it. Farming microgreens is a major area. We recently studied a paper saying that a person is earning 5 lakh rupees with just 10 cents of land with microgreens only. He supplies microgreens to the leading this one 5-star hotels and gets around 5 lakh rupees. So hydroponics, aeroponics for exotic vegetables and fruits, people can look into the aspect of it. After studying, you can make these things, and functional foods is one of the area where you can get yourself equipped. And another one is also having a plant defensin. Plant has got a single small 23 amino acid protein called defensins. It is like an immune system of the plant. The defensins are present in major radish seeds and a lot of leguminous plants. The defensins are known for protecting the plant from infection from bacteria and fungus. Can we use them for our benefit? We have to ask this question and we have to look into it. Functional foods, yes, probiotic, prebiotic, fortified, dietary, omega-3 rich fatty acids, spices, plant sterols, so many things are there. So these are all these industries. If you make a startup after your studies, you can make inroads into it. Finally, before closing it, yes, I think I'm going to close it. Diseases can be cured. Disorders can only be managed. In short, the one you get sick because of the external can be cured. The one which got yourself sick because of your internal problem can only be managed. Like diabetes, blood pressure, anything can only be managed, but infection can be cured. Yes. If you have questions, I'm ready to answer. தமிழ்லையும் கேட்கலாம் சொல்லுவேன். If you are a little bit hesitant. Yes, sir.
The same kind of question as we talked about in the morning. When you have a formulation with several compounds, is there a specific way each of them, most of them, participate in something to resolve the issue? And what happens to the remaining? Would they cause any other adverse?
This plant system, if you take it into this, it's been long since we've been in front of our human system, because we have been taking long ago. I am not finding a plant which is exotic. I am finding a plant which is being taken by us every now and then or every day. I am only concentrating that plant compounds and extracting it making it into a formulation so that it is effective. There is one thing called, I have forgot to mention in the morning, I was speaking to him also. It's called Anubanam. Have you heard about Anubanam? Nothing. Anubanam is from Ayurvedic science. See, that is a beautiful thing. You should give the medicine along with coriander. If you have the problem of liver, then you should give medicine with milk. There is something like that. This is called Anubanam. Neyoda sendu kudupanga. You have to give it with ghee. You have to give it with milk. You have to give it with water. You have to give it with hot water. Why do they say that? There is a reason for science is there. Because it brings that compound to the site of action. It will be more prominent. So the compound which need to go there had to be provided along with that. When I see some formulations of Ayurvedic system, I find a certain plant material which is not going to be useful for asthma at all. Why do they put it for asthma? When I start questioning that, then I found that that plant material has got a different function which can relate it to asthma or to the site of action it brings in, or it potentiate that. For example, curcumin, if I say it is from turmeric, you all know about it. Do you know that presence of piperine, that is from Piper nigrum, that is made of black pepper, it increases the potency of curcumin 1,000 times. Like he said, saliva, the presence of saliva increase the infectivity ratio. Same thing happens there. The presence of one particular plant will potentiate other plants. So you don't have to give curcumin that much. You can give less and more effective. So it is all permutation, combination. See, there is a big advantage in plant system is it is not mostly toxic. Mostly. I don't say it is non-toxic. I will say it is 95% or non-toxic. They don't have any other interference with any other drug molecules. 5% are there. We have to identify that. That is there. But these things are not. We have to see the literatures. Because these literatures are available nowadays. So you can find out from the literature whether, for example, St. John's wort, which is known to interfere with blood pressure. You have to ensure. For example, if you are taking clove, clove is nothing but a dilution of blood. That is, it will thin the blood. So when you are taking aspirin, you should not take this blood thinner also in a higher quantity. That is not possible. You have to ensure these things happening. You have to see both science as well as traditional knowledge has to be blended in support of allopathy system. I am not against allopathy. I am very clear. I am only supporting allopathy. How it can be supported? It is only a targeted therapy. It is a whole therapy. It is a whole system therapy. So I am only trying to correct it. For example, sarcoidosis, I told you, one of the person who has got sarcoidosis in her liver, that is clubbing of cells in the liver tissues. The liver cells clubbed to form like a cancer, but it is not cancer. It is not a cyst. It is clubbed. So it is an inflammatory reaction which happens. So what happens is when anti-inflammatory medicine is given, along with liver rejuvenation, because she was taking tuberculosis drug. So it bounds to, rifampicin is bound to attack that liver system. So when she was having her bilirubin content at 3, I suggested her to take Li52, along with that my anti-inflammatory drug. So Li52 corrected her, now it has come to 1.4 from 3. About last 6 months back she had, and now it has come to 1.4, which is very closer to the normal, and still she is not normal. But she is continuously taking her medicine of TB, antibiotic regime is going on. So continuously she is taking the Li52 as well as my drug. So I give my drug free of cost to anyone, whoever it wants it. If it is probably that blood test result I am seeing it, I used to give the drug at free of cost. Even the courier charges I don't get it. So I ensure that the person is giving the proper result to me every month, so that I follow them very properly, whether the drug is doing well with them or not. So that is how. So that is the reason. I don't know how it acts. Still now mechanism of action, if you ask, no idea. I don't have. But I am sure that my drug is a safe one. I am sure that it can go with any other drug, but if it does good to you, that is what I wanted.
Good evening, sir. Thank you for a valuable presentation, sir. Sir, actually, I have a statement. I want to know whether it's a myth or a fact, sir. Like, it is the assumptions of most of the people from whom I have heard, sir. Don't approach naturopathy if you are at any stage of a disease, like cancer, joint pain. Most of the old age people will get. Most of the suggestions would be take allopathic medicines because you have crossed a certain stage, and also naturopathic medicines will take more time to cure your illness. So better you opt for allopathy, like that, sir. So I want to know whether it's a myth or a fact. But you have many examples of curing the final stage diseases, as you mentioned, for lamb, eczema.
It is not final stage disease. I am sure that it is not a final stage. They have lost hope.
Like
They have, that is the
conditions.
allopathy people, the doctor. I can tell who's which doctor, which hospital, and everything. I don't want to do that. But they have certain limitations. See, all doctors, I am not blaming allopathy system at all. I am only saying, along with your allopathy system, along with sirolimus, that girl was taking the drug. I have not asked her to stop sirolimus. I have asked her to stop sirolimus after she got her X-rays clear, thoracotomy taken away, her walking system, 6 minutes walk, when we measure it up, she was only walking 110 meters for 6 minutes. Her normal walk should have been around 500 meters. After she came to 520 meters only, I asked her to stop my drug. And after that only asked her to stop the sirolimus. So I don't say that sirolimus need to be stopped. Instead of that, my drug to be taken. I never say that. Or even allopathy system cannot be. Along with allopathy system, you can give Ayurvedic system. Where you have to stop it, where you have to stop that. You have to know.
Yes, sir,
That's
but
all.
these are not assumptions of like doctors or any physicians.
It will not
sir.
be. It's obvious,
people,
no?
normal
Yeah, they don't
people,
know this science.
Like they don't know, they don't
Yeah,
know about these
that is
evidences. Like, so I asked, sir.
Yeah, correct.
Because many
But not only
suggestions
that. And the second point is, most of the doctors, they only practice, they don't read. They
yes,
only
sir.
take pharmacies, pharma companies who are talking, who are giving to the doctors, this will do that, that will do this. Based on that, they give prescription. See, I know a COVID patient of my own brother-in-law, when he had this one, when I gave him drug, he was against it because his neighbor was a doctor. Here in Pyramid only, he is staying. His neighbor was a doctor. His doctor has suggested him to take steroid. I asked him to not to take it, but he took it. He was admitted in the hospital. And what happened is, after that he came, because I asked him to take my drug during the hospital time itself, given him sideways and this one, because hospitals were not allowed to enter anything, because I know certain people in the hospital, so through that it has been given. And finally he took that medicine and came out, because he was having, one stage he was 50% SpO2 level only. He was brought back. And after that, when he came back, he was given a drug. Immediately he contacted me, this was the drug he was given. I told him not to take that drug, because that drug is given for transplantation purpose only. I have told him not to take that drug. I have sent him the publication of that drug which has given that, because there is no need for it. This will do harm rather than doing good to you. So I suggested them. Whether to follow it or not is up to them. But I suggested him, he didn't take that drug finally. It's an anti-cancer drug, so not that it was an anti-cancer drug, it was suggested. Because whatever the pharmacy company says, when I searched in the internet, that was the anti-cancer drug was given for COVID patient in a particular country it was given. So I suggested him, it was still in the infant stage, they are introducing it because of the COVID. COVID was so much problem, they wanted to introduce it, any drug. So that was the problem, please don't take it. That's what I suggested him. He is doing good now. So that is why I am saying this is, doctors are not familiar with allopathy, Ayurvedic drugs or plant-based drugs, first point. Another point is, They don't want to try anything because tomorrow they will be in. The Supreme Court has already told very clearly that allopathy doctors should not prescribe Ayurvedic medicine. Ayurvedic medicine doctors should not prescribe allopathy medicine. So it has clearly said it is against legal, so that they cannot do it. They can only suggest them without any prescription. No prescription can be made. That is why they don't do that. In hospital, they don't do that. When you go to the clinic, separately they put it in the paper and give it to you. For example, Bonison, one of the drug, which is given, you could have all been taken, Bonison. And because earlier days, after childbirth, Bonison is given to any infant for bone strength. But Bonison is from plant. It was prescribed by the doctors. But after that, they didn't prescribe at all. After that ruling came, they didn't. They cannot. That is why they don't do that. They don't know this sort of science which is happening. If that is the reason, there should be a marriage between these two to get a better result for the human beings. There should be some understanding. Both allopathy as well as Ayurveda understand the science better and for the betterment of the human being. This is my view.
Thank you, sir.
Plants continue to be an important source of inspiration for drug discovery. You explain the need of plant-based drugs in inflammatory disease and antimicrobial drugs to the society. Thank you, sir, for sharing your knowledge and expertise with us. We look forward to applying the insights gained from this session.
Learning is the product of active engagement and participation. I would like to invite Dr. K.M. Saktivel sir, Assistant Professor, Department of Biochemistry, PSGCAS, to introduce our respected resource person.
Good afternoon to all. It's my great honor to introduce Dr. G. Sridevi, an eminent scientist and educator in the field of plant biotechnology. Dr. Sridevi is currently serving as Assistant Professor at Department of Plant Biotechnology, School of Plant Biotechnology, Madurai Kamaraj University since 2010. Madam's research expertise include plant molecular biology, functional genomics, RNA silencing, and plant genetic engineering. With over 2 decades of academic and research experience, Madam has significantly contributed to genetic engineering for biotic and abiotic stress resistance in crops, biofortification, and metagenomics. She holds PhD in biotechnology and pursued postdoctoral research at prestigious institutions such as University of New Hampshire, USA, and South China Agricultural University, China. Dr. Sridevi has mentored several PhD and MPhil students and actively involved in pioneering research projects, which is funded by DBT and UGC of more than 5 crores. Madam has several significant publications in high-impact journals and also contributed to chapters to internationally acclaimed books. She is also a recipient of several accolades, which includes CSIR Senior Research Fellowship, and she is a proud life member of Society of Biological Chemists and an active contributor to national and international scientific communities. Madam is also involved in a task force member from DST. We are privileged to have Dr. G. Sridevi Madam with us today. I extend a warm welcome as she enlightens us with her talk.
Thank you for your wonderful words, sir. It's time to honor Dr. G. Sridevi ma'am. I would like to invite Dr. D. Victor Arokya Das sir to honor the resource person.
for psoriasis?
Erythrodermic psoriasis, just now I have formulated a
Yeah, yeah. This one is, I don't know, he has
both external as well as internal.
external.
Am I audible? So good afternoon, one and all. First, I would like to thank, it's my parent institution. I am back here and I'm happy to see all my professors, lecturers here. And today, like, I feel very energetic, right? Go home. That's what I felt. Actually, I will say this is the first stepping the stone for my success. So PhD has been one of the ladder for me. And today I am here because of the foundation from here. And first I would like to thank the institution and also my parent department, Biochemistry. And those of you who are studying here, so you know the current batch of faculties. So, but we have been associated with the earlier faculties. So I came to know like last week, Brinda ma'am got retirement. So we are very closely associated, and we had alumni meet also 1 year back. So we were here in the campus. So it is nice to be here again. And I should thank my professor, Victor sir, who was my guide in my UG and PG. And today I'm happy to get the memento also from him. Thank you, sir. So today what I want to, so when he called and asked me, you just present like your part of your research. So what I thought is, like, because the students in your age, like UG and PG, when we people come and deliver lecture, so earlier I was also sitting there. But what is the main purpose of delivering these kind of lecture is that we want to initiate the spark, okay? So when I was a student like you, the spark was initiated by one of the student, one of the speaker here. That is how I continued my career as a PhD, postdoc, everything, and now I'm into the research field. So at least today, whatever the talk happened by the professors, given by the professors, we hope some spark we would have initiated, okay? So those who got the spark, like to just take it as a touch in your career, okay? I hope whatever the talk I'm going to give you will initiate the spark in you. So today what I thought is, like, since my area of expertise is plant biotechnology, so I would like to give a brief outline. So I don't want to go very deep into the research, like in depth, but an outline and also covering the depth of the research what we will be working. So I gave the title. Okay, so we start from genetic engineering. So it started like 20 decades back, and now we are moving ahead into the digital revolution. So all of you must be aware of the word artificial intelligence, right? So how this artificial intelligence is taking, like, stepping the stone into each and every sector, okay? So I thought this will be helpful and it will initiate and it can guide you people into the future technology. That's why I gave this title. Can you go to the next slide? So if you look at all this research, what we people are doing so far, especially related to agriculture biotechnology, what is the purpose? There should be some purpose, right, when you want to do a research. So the purpose of our research, especially in agriculture field, so the main thing is to provide food for the human population. So if you look at the slide here, can I get the pointer? Center. On பண்ணிட்டீங்களா? No. ஆன் பண்ணலையா? Okay. Okay. Thanks. Okay. So if you look at the slide here, so this is in 1900. So in 1900, whatever the production they had made, the farmers, so each farmer, whatever the production they had made, they will be able to feed 10 people, okay, whatever the rice or wheat, whatever they produce. And if you look at after 50 years, so each farmer, they can feed 27 people. You can see how much the production is increased. And in 2000, he can feed up to like 130 people. And later, like now you look at 2030, so we have to feed more. Why we have to feed more people? Because the growing population, okay? The population is increasing day by day, and also there is a need for the production day by day. That is the purpose. And they wanted to do all this modification, they want to increase production and all those things. So I will come to the slides later. So if you look at the map, WHO has released hunger map, like how many people are hungry in a country without food, okay? But you could have remember the word or saying of Bhardiya, right? What is that song? There is a, not Achamale. All the people know Tamil, right? Tani oruvanakku unavene illenil. But you can see what is the status of India. So you can see the color. So it is like orange color. You can see how much percentage of the population doesn't have one meal. Even a meal in a day is they are not getting this much amount of people are not getting a single meal also, okay? So this is the situation now because of the increasing demand in the population and the productivity is less. So if this situation continues, so what is the challenge we have? By population in 2050, we would have reached like 1.5, more than 1.5 billion. So what happens? The shrinking of cultivation land shrinks, there is a water scarcity, and also malnutrition, deterioration, and also the soil quality decreases, automatically the production decreases, and now the biggest target what we are getting is the global warming, right? So everywhere all the transport, everything is increased. So they are now, yesterday I was just seeing some of the article, by 2030, the world will visualize the first Arctic Ocean without ice. That is what scientists are expecting if this climate change continues. So this is the challenge we have in front of us. So in this scenario, what is the thing, how agriculture is going to have an impact? That is what, and what all the people as an individual and as a scientist, what we have to do. That is the thing today I'm going to talk about. So what is that, my talk will be just, I'm going to start from the traditional plant breeding, moving into genetic engineering, then deep into the transcriptomics and other thing, and also how it is integrating with artificial intelligence, and the future of agriculture, what we are talking by 2050, we are going to step into the precision farming. Okay, I'm going to talk about all these things. So the last part of my talk will be talking about the artificial intelligence and precision farming. So all the people know what is plant breeding, okay? Especially the agriculture students will know much better than us. So being a science student, you all have read what is plant breeding and what is selective breeding, what is advanced breeding. And if you look at the domestication of the crops, it has started way back 10,000 years ago, where people started like crossing two plants for the different traits. So they want to have the trait of this. So if you have a plant with all starts with the Mendelian age, okay? You must have studied Mendel laws of segregation and all those things. So from then onwards, this breeding, everything has started, and many progress have been made. And today I'm going to take one example, okay? So this is the research which we are currently working on. So we are working on the sheath blight disease in rice. So this particular disease is mainly caused by the fungus, Rhizoctonia solanae, okay? And this is the second fungal disease next to blast in rice, and it is one of the serious threatening disease in rice. And the yield loss is very wide. Almost 45% of the world cropping area is affected by this disease, and there were like $1 million loss happens. So if you look at the breeding aspect for this particular thing, there is no resistant rice cultivar available for this particular disease, even though we have some partial cultivars like Jasmine 85 and Teaching. Okay, these are some of the Chinese rice varieties and Japanese rice varieties. They show some partial resistance, whereas we don't have any resistant cultivar. So breeding is very much difficult in this case. So we have to go for another option. So we automatically move to the next option. So we went to the second option. So it is a step ahead, that is genetic engineering. So genetic engineering is an extension of the plant breeding. Okay, so in the plant breeding, what happened? You have a donor plant and the commercial variety. The desired gene from the donor variety will be transferred into the commercial variety. That is what happens in the breeding. Whereas in the case of genetic engineering, you have a donor with the desired gene. So what happens? You have the desired gene from the donor is removed, isolated, and this desired gene is introduced into the commercial variety by transformation, and the commercial variety, improved commercial variety with the desired gene. So this breeding want to go for like 5 to 7 rounds of cycle reproduction. So finally you get the desired variety, where here with a single cycle, okay, single cycle you can get the improved commercial variety. And this gene, whatever the disease, whatever I am telling, so this is mainly caused by the fungus, the Rhizoctonia solanae, and this is the symptom of the disease, and this is the life cycle. So it is survives in the soil when, as you all know, the paddy field is full of water, logging area. So this sclerotia, when it survives in the water, when the water comes in contact with the paddy plant, it tried to invade the paddy plant and produce this disease, and finally it affects the whole plant and finally results in the death of the plant. So that is what happens with the things. And next question can come. So when any pathogen attacks plant system, will the plant defend it or not? Plants have their own system, like defense signaling system. There are many defense signaling pathways involved in that, and all these things you must be studying in your plant biotechnology classes. So I am not going into detail. So the ultimate final thing I want to highlight is, so all the defense genes will be activated as a part of defense signaling cascade. So we are going to use these genes and try to overexpress and utilize this for the transgenic plant development. So in the typical transformation system, we try to identify a rice plant. We always lay a platform for the tissue culture thing, and the transformation protocol, everything is laid out very clearly. And we used to go for a strategy, the antifungal gene, that is a pathogenesis related gene, either single or alone or in combination. We have tried, and we had developed the transgenic plant harboring these genes, and after developing the transgenic plants with these genes, we tested again with the fungus. But with the single gene, we were able to get some 20 to 40% resistance, and with 2 genes, like a combination of genes, the resistance from 40% increased to 60%. So maximum the resistance we got with the genetic engineering is 40 to 60%, but we couldn't get 100% resistance. So now we had to move for the next, the technique or technology to go for a complete resistance, or the way how to protect from the pathogen. So there come in, okay, the next one, what we call as RNAi. I hope all of you must be aware of this word RNAi, that is RNA interference, or you must be familiar with these people, right? Do you know who these people are? Who got Nobel Prize for the RNAi, right? Andrew and Fire. So they got the Nobel Prize for the RNAi, and after the discovery of this one, now they wanted to look into the RNAi pathway in the plant system, and also we have I'm not going into the pathway in detail, okay? And you can see there is RNA interference happening between the kingdoms, so between the plant and the fungus. So we can very clearly use RNAi mechanism. We can target the host candidate gene, and also we can target the fungal candidate genes. So first we should know which are the host genes are trying to interact with the fungal genes. So first we should know what genes are playing a role in the plant pathogen interaction. So for that thing we should first clearly understand the transcriptomic part. So towards that what we did is, so we did a time course experiment to identify the candidate plant genes and also the candidate fungal genes which will interact with each other in order to increase the disease. So what we did, so we infect the rice plant, all these things are done in the control condition. So we infect with the pathogen, and you can see here, so with 12 hours, we did a time course experiment from 12 hours to 72 hours, and after different time point, we harvested the rice plant and we subjected to the NGS, next generation sequencing system, and a complete transcriptome profiling has been done using the thing. And the platform what we used was the Illumina or Solexa platform, where we did the NGS thing. So where this is the outline for the NGS thing. So they isolate the RNA and it was converted into the cDNA, and the ligate, the adapters to that, and completely the sequence the first end and also the second end. So you get two sequences for the paired end sequence, and before that they actually check the RIN value of the RNA, like how good is the RNA, and the RIN value should be greater than 6. If the RIN value is greater than 6, the RNA is ready for converting into the library, and they make a library and do a complete NGS analysis, and after the analysis you get a raw reads. Okay? So the raw reads will be given in the form of the FASTA. So it will be something like 2 to 3 GB. Okay? The data will be given here, and so this is the raw read what we get, okay, at the end, and in each case you will have like 2 to 3 replicates. So the biological replicates and also the technical replicates. So in that case you will be having each sample, you will be having 2 reads. If you have say C1 is the control, so you will have R1 and R2. Similarly, you will have the biological replicates, the control 2 and control 3. So similarly for the different time points also we had the biological and technical replicates, and totally we had something like 18 libraries made out of it, and these 18 libraries, each library had generated a raw data something like 2 to 3 GB, and finally we took all these data into the analysis part. So what we did, first you do a quality check, okay, with using the fast QC method, and you do a quality check, make sure the quality is good, everything, and finally there is an adapter, right, for sequencing. So you try to identify that adapter, you remove the adapters using the trimmomatic method, and after trimmomatic you check for the quality using the fast QC, then finally you map it, okay, either with the reference genome or de novo assembly. Here since it is a rice, already we have the rice genome assembly is there, ensemble assembly. We try to map with the reference genome, and finally after making a mapping, then we subject that mapping part into the differential sequence analysis to identify the differentially expressed genes during the pathogen attack, either upregulated genes or downregulated genes. So this is what we have done, and finally we make, in the case of to identify the fungal genes, instead of reference based assembly, we did a de novo assembly and we subjected that to Trinity, and we removed the dependencies. Finally we did a blast to grow analysis to identify what genes are involved in what fungal genes are involved in the plant pathogen interaction. So this is a summary of the raw reads what we have from the 18 libraries, and this is the first set, I said the quality check, right. So this is before trimming and the raw data, the top one, and here after trimming you can see this redundancies all removed in this case from the raw data, and you can see a very clear data coming out after trimming. And after that we go for the reference based assembly, and now where we removed the rRNA sequences and other thing, we try to map with the reference genome, and we can see 97 to 98% mapping with the reference genome. And here for mapping we used Orisa Sativa ensemble database, that is reference genome 46. So we did a complete mapping for this thing, and after mapping we tried to align those things with us. This is the summary of the aligned data what we have with the ensemble database, that is ORISA Sativa, we could see almost like 98%, 97 to 97%, 98% alignment has been done. And after this, we try to go for the differential sequence analysis. One is using DSeq, another one is the cufflinks. So of course, we just comparing two different softwares to see which one is working better, and we found that. So DSeq2 is always better than here. And after identifying, so after identifying the number of upregulated and downregulated genes, we did a volcano plot to just look at the top 100 genes in how much category they had fallen, and whether these genes, whatever gene that is present in 12 hour, whether it is present in other time points also, or whether the particular gene is present in all the time point. So all those things, we had just a complete study. We had done a volcano plot of the top 100 genes, and we did a heat map analysis along with the control. So control versus 12 hour, control versus 24 hours. For each time points, we did a complete heat map analysis, and we were able to identify the top 100 genes, which is upregulated and downregulated. And finally, we did the GEVO analysis of the top represented genes, upregulated and downregulated, and we could see where these genes were fall into which category we were able to identify, and this is what it is plotted here. And here finally we had a Venn diagram to show a combination of the top 100 genes, so whether it is present only in 12 hour or it is present in 12 hour, 36 hour, 48. So we were able to categorize all those genes, and we were able to narrow down those genes which is present in all the time points or few time points, whether at the early time point or at the mid time point or at the later time point. So in that category, we were able to get all these genes listed here. And finally, we did a expression analysis also for those genes. So this is the upregulated differential expressed genes, what you see here. And the next one is the downregulated genes. What you have here is a Venn diagram. And finally, we had taken just a candidate selective genes from each category, okay? And we had seen the log, the fold change, the expression level. This upregulated, this I have shown only for the 12-hour data. So this has been done for the remaining time points also. So here I am showing the data for only the 12-hour one and the downregulated for the 12-hour. So in each category we have shown all those things. Now we have identified the candidate fungal genes and the plant genes, so which are playing a major role in the plant pathogen interaction. So what we go for the next one. So when in the reference base assembly we were able to identify the plant genes, and in the de novo assembly we were able to identify the fungal genes. And the next one what we need to move is the RNAi mechanism. So which I am not talking here because that is another big part of the work. I am just briefing it now. So we have now moved using the candidate fungal genes. We used to make a double stranded RNA containing vector. So where it is a RNAi mechanism. So we just transform into the plant system so that the plant can synthesize its own double stranded RNA, so that in the fungal pathogen attacks the plant system, the plant can protect itself from the fungal pathogen. So already we had done initiated all those things. The final evaluation is in progress. I am not going into the detail of that one. So this is with regard to the genetic engineering and the RNAi mechanism. In the meanwhile, now comes the next technology what you are currently having. So all these evolution, what say from the breeding, genetic engineering, RNAi, all these things is not a single year I am talking about. This is something a 20-year kind of a thing, the evolution I am talking about from where we started. So when we started doing a PhD, that time genetic engineering was one of the biggest active, like attractive area of research. So most of the people, like by 2015, 2000 to 2015, you can see many papers on transformation, overexpression, genetic engineering, development of transgenics with this gene, that gene for the biotics test, abiotics test, all those things are coming up. And after the RNA mechanism had identified, people were narrowing down to which genes are playing actively or interacting with each other component in the case of disease like biotic stress, or in the case of abiotic stress, suppose in the presence of like cadmium or iron. So how whether it is a toxic. So, or what are the genes that is playing a role in the iron uptake mechanism, zinc uptake mechanism. So, those things were narrowed down after the NGS has been developed, and then they started targeting those genes to either they want to downregulate or upregulate. So, those mechanism and all those things they have started, and we have moved from the genetic engineering to the genome engineering. Now, people are working on the CRISPR-Cas system. So, I have not told about these things here because it is yet another big, it will take another 1 hour of the talk. So, I am not getting into the detail. So, now people have moved from genetic engineering to genome engineering. So, now they are advancing with the CRISPR-Cas and all those things. Now, in parallel, we have another technology that is coming along with the thing is the digital revolution. So, now we have all these techniques developed. The breeding, genetic engineering, genome engineering, you have target everything. But if you look at the other side, this is in the scientist part I am talking about. If you look at the other side, how will a layman know about all those things? The farmer should know what is the disease. Okay? With all the innovative technologies coming up, so is he equipped with the latest technology? No, still not, no. We are not very clear about all those things, but we should develop all these techniques which is very easy to him. Okay? If you talk about 10, 20 years back, do you think you can have an Android phone? No, right? But even I can say today, a beggar in the signal is having an Android phone, right? Whether he has studied, he is a literate or illiterate, it does not matter. But the person knows to operate an Android phone. Okay? So, we should get that kind of a technology to a farmer. So, that is where we are now getting into the digital revolution. So, the farmer should know what is happening in this field, and all those things has to be given him. Okay? This is what we see earlier talk, what I have been talking about is the science. So, whatever the advancement what we are doing, and this is coming to the application. So, now this application and science I am going to finally link later. So, this application what we want to do is how we have to get that in the form of application to a layman. That is why we are getting into the digital revolution, and earlier people don't have laptops and all those things, but now even a student without laptop cannot enter a school, right? This is now it is going, the technology is going so fast. So, earlier people were working with all the, if you have seen the old agriculture thing, they must be having a cow and all those things. Now they move to tractor slowly, and all those things are advancing, right? Like how this is also going to advance. So, now we are in the digital era, and what happens in the digital era? So, with this disease what I am going to say is, so how we can identify, how a farmer can identify this disease. As a layman, how can he identify the disease? So, the technology should be given into his hands. So, for that what we try to do is, so we had taken the photograph of the disease and all those things at different time points, and we have been recording all those things. So, we were trying to take all those pictures and we were converting that into a hyperspectral model. So, that is what you, when you say a normal picture, it will be like colorful picture. But if you convert into it is a simple RGB, whether the picture is there or not, it will be present or not present, something like a gray color, RGB format it has to be converted. So, we have many softwares for all those things, and we were trying to convert whatever the pictures we had earlier, whether that can be converted into a hyperspectral model. So, one of my students, he attempted, he is very good in computer. So, he attempted, I asked his questions, he said I will try this, and he attempted these things, and that he made with one of the system, that software that is Pix2HS. So, he were able to convert this into the RGB format, and he was able to generate the things. I asked him, because we were working in the genetic engineering, I was asked him like whether you can say whether you have a control plant and a disease plant. Is there a way to discriminate? And this model he was able to discriminate and the other thing. So, we had a different time points. We had a photograph of this. So, using this time he had converted that into the hyperspectral data, and that hyperspectral data he made it a hyperspectral signature, And finally, when he got this progression profile, so yes, it clearly matched with whatever the data we had as a plant disease progression. So it was very clear when he did a complete, he used the software and he completely matched everything. Finally, he got it as a value, and that value, when he plotted as a graph, finally he got this kind of a graph which clearly matched with our data. Okay, so we thought, okay, this can be done. So the next thing, we wanted to develop our own model. So all these softwares are already available, but when it comes to artificial intelligence, it is a neural network one. Either you have to go for the CNN model. So we thought we can go for the CNN model. So what we did, so this is how, when the picture is taken up, when you develop a model on that, it is going to make it as a segmentation. Each segment will be identified and each segment will be fed into the computer as values. Okay? In that case, when you have a clear leaf without any disease, that will have one value. If it is a diseased leaf, that particular segment will be given another value. So that is what, so one of the MTech student, who is an MTech student in data science, that student contacted me for some of the project. So in biology kind of a thing, I asked the student whether you can make like programming kind of a thing. That student said yes. So I asked that student to make a program for the thing. So they developed their own hypothesis and modeling everything. So we developed a software kind of a thing. We were able to make this thing. So I didn't put the things here because we want to go it for patenting. So I just told what is the basic here. So we were able to get this model out of that particular thing using the CNN model. We were able to develop the software, and that software is now we wanted to, at least the next part of the thing is that software, if we can given like utilize to the farmers, whether the farmer just with a simple Android phone, if the software is also installed in that, the farmer when he displays the phone or the camera over the thing, the camera should be able to tell him with the software whether that particular plant is diseased or healthy. Okay? So this kind of the thing, now we are in the process of the thing. So slowly it is going to be patented. And the next thing what now we want to get is, now slowly people are working on this kind of a thing. Slowly the whole farming area is going to be under the control of a computer. Okay? So you start from the water irrigation. And I know many fields in and around Coimbatore, you have this irrigation control coming with the computer. So with, they farmer may not go to the field to fetch the water. Just with a simple Android touch phone, he can just fetch water. So which area of the land he can fetch the water, which area of the land he don't want to fetch, he want to stop it. So he can close or open the walls with the help of the computer, the Android phone and software, so everything is available in the thing. So that kind of a system is there. And slowly the Agriculture University Extension Center, they have developed this monitoring the soil health. Every now and then they used to take the data, the soil samples. Those soil samples from that particular latitude and longitude, they used to put it in the website. And I hope all the people must be aware of the PM Kisan, right? Pradhan Mantri Kisan. So the thing have come up in the app format. So that app, all the farmer has to be registered. So when they go into the app, so if suppose this particular area, what is the status of the soil, nutrient, all those things, they can check with that thing. So based on that, it will say this month, this is the nutrient status of the soil. So next month, you people have to, this is very less, this has to be added. So automatically he gets this kind of information in the app and also the WhatsApp. This is what is going on. And next thing is, now how many of you have seen, like if the farmer wants to just like spray medicine, whatever the pesticide or something like that. How many of you have seen how they used to pesticide? Anyone can tell? They used to carry some kind of a tin with the spraying machine. Okay? Now how many of you know that drones are being served to the farmers for spraying pesticides? Now the Agriculture Extension Center is doing that. the drones are given to the farmers. They can hire the drones, they can spray the fertilizer. So these kind of you fetch into the drones can identify which is a diseased and which is a non-diseased plant. So since we have at the different time points, so they can detect the disease very early in the thing, and the remedial one, whatever the genes and all those things which we are working in the future, that can be served as the remedial part. So this is what many people are now working on, and slowly you can see the agriculture field is slowly getting into the digital. So they are using the digital part here. So now you have the sensors, connectivity. So all this data, everything comes into one simple phone, the automation. So if this continues, what will be the future? That is the question. I am leaving it as a take-home message. So this is how it is going to be in future. You will have all smart robots doing all kinds of things, identifying, okay, each and every part of the thing. Okay, this is what is going to be in future. Thank you. Any questions? No questions? So the talk did not initiate any spark. Right? If there are no questions, that is what we feel. So if the people have understood, they will ask questions. If they don't understand, they will not ask questions. So what message should we take? So any question, you can ask, feel free to ask, because this is the platform that you people can interact. So when you ask many questions, you will find many answers for the questions. Yes, sir.
It's going to work probably very well once you have enough data. If you look back in science, we have been publishing only positive results. We never have any negative results published.
That's true.
For AI, it requires both positive results and the negative results to kind of understand which one is right direction to go, which one is the wrong direction to go. What is your thought on what we should do, in terms of, should we go back and get all these negative results, somehow publish, put it in a database or whatever? Because unless it's in the cyberspace, AI is not going to work.
I understand.
What are your thoughts?
So in my perspective, after entering into this particular field, what I felt basically is, so it is trying to mimic a human brain. So when a brain, they are trying to mimic, but I don't see 100%. But a human brain, what it sees and what it does, it judges everything, okay? And we want to train a machine to do that. But in like what we are doing, we can understand and do. But making a machine to understand, I think some parts of the thing we are still lacking. We are still in the preliminary infant stage, I will say. This AI is still in the infant stage. It has to be explored more to answer your question to that part.
See, in case of humans, we already have some things. The experiments, you had some failed results, negative results. You already have it in your mind, hey, that one is not good, don't do it. You already tell your students, right? So that one is very clear. Because although you have not published, you have that information with you. Whereas for AI to work, everything needs to be in the cyberspace, in the data somewhere.
Yes.
In your case, you have the data. It's not published, but it's in your mind.
Yeah.
So that's why I'm trying to understand how would that happen, because we have lots of data
That is true.
in the last 50, 60 years or longer.
And whatever the data, whatever the software they generate, only those data is in the cyber, as you said,
Correct,
those
correct.
kind of a data is available in the as a software, as a program, as an algorithm, it is still there. But for the data, as like say negative things, what we have, I think we need to explore more. As of now, I don't have answer, honestly saying, but we need to
Just
explore
out of
more.
curiosity. Yeah.
No, at least you throw a question, I will take your question. I agree with that thing.
I have a question. See, thing is, now that rice sheath rice sheath disease, in that, did you find any expression of genes different from infected and uninfected plants?
Yes, we had a very array of genes, especially those pathway of the things. So I haven't got the slide here, that is in the publication part. So we have got an array of genes, especially the PR proteins, and not only in the PR1, and also some of them the signaling pathways. We have identified those genes and we have validated the things. So we are now taking those genes and getting into the RNAi. So we are moving for the genome editing part.
One small question. Yeah, it was interesting. You said you have some kind of gene signature for either promoting or resisting the resistant property or susceptible property, whatever it is. And you have only dealt with siRNA, correct? Or
No, sir.
RNA
I have
silencing.
only RNA silencing mechanism.
Yeah. Because I am biased towards microRNAs, I am tempted to ask this question.
Yeah,
Have you looked at any microRNAs that could do, because they can actually attack or silence multiple mRNAs.
Yes, I agree with.
Theoretically speaking, it should also to look at microRNAs. And are there any data or are you looking at it?
When we did the mRNA sequencing, parallelly the same 18 micro libraries, we are, so my collaborator,
Looking at it.
okay, Dr. Usha's student,
Yeah, Usha, yeah.
okay, her student, Dr. Gopal, who is a collaborator of mine, so he works on the microRNA part.
Okay.
So he had completely taken care of the
it
microRNA
would-
libraries,
it would be
and
interesting.
he was able to get some of the microRNAs. So I am looking at the mRNA, he is looking at the microRNA. So we were trying, now we are now next level, we have to go and link the microRNA and also their target in the mRNA. That is what we have to move to the next phase.
In the plant system, they have 100% complementarity.
Yes.
So that also makes it more interesting.
Yes.
Thank you.
So he works completely on the microRNA. So that part is taken care of by him. Yes, most welcome.
Okay, sorry. I've noticed a lot of things in DEGs, differentially expressed genes. When you put the volcano plots, you put 2-fold, 5-fold, 10-fold, what. But if I'm going with the thing where Lakshmi asked you this question, if it is 0 in the healthy or infected, so when the infected has anything, it will be infinity, because right? So how do you deal with such numbers, 0 and infinity, even if it is 0.1?
No, actually when the data comes out, finally we get into the logarithmic first, the data. And in that case, whatever you say that 0, those things will be coming out as a base thing. So don't we always keep some value as that when we give an input for the particular analysis, so we always give some kind of a window for the analysis.
The question is different. For example, let's take ABCD. ABCD is not expressed in the healthy individual, but with the infected one is expressed 1, one is expressed 10, one is expressed 100. All of them will be infinity because the bottom is 0.
I got it.
How do you deal with it is the question.
So when we pull all the genes together, so we look from the top one expressed to the one which is expressed, say, as I say, the 10-fold, we get it first. And the later the 2-fold, 1-fold, and all those things. We will be looking at that. Infinity fold, then that will be more. No, when that is what when I said the top 100. So from the values, the topmost values we narrow down. So where the values are more, those we take it first. So those are the top priority one. Those plays a major role. That is what we take it, the things. But I feel still it has to be explored more, because when it comes to the research part, we are very strong. But when it comes to computer part, we are still learning only. That is what I feel.
Systematic, fantastic.
Thank you. Thank you.
Recognize your done a very good job in logically going through the problem. I appreciate it.
Thank you, sir. Thank you so much. No more questions? Okay. Thank you so much, and thank you for giving me an opportunity. And I also thank my professor, Victor sir here, for arranging me to give a talk and to have a discussion with all of you. Thank you so much.
Thank you, ma'am, for your enlightening words in the field of plant biotechnology and involvement of genetic engineering and AI in plant-based researchers. Your knowledge on antifungal plant development and transformation of RNA inspired us. As the saying goes, when we feed others, we feed our future. You have given us a vision for a better generation. Thank you, ma'am.
Validation is about recognizing the contributions and achievements of others. Let us acknowledge and celebrate the ideas exchanged and the milestones achieved throughout this conference. I would like to welcome our Principal in Charge, Sir M. Sengatuvan Sir, Associate Professor and Head PSGCAS, Professor Kinney R.M., Emeritus Professor, Department of Biological Sciences, National University of Singapore, Professor Devarajan K. Arunagaran, Emeritus Professor, Department of Biotechnology, IIT Madras, Chennai, Dr. Lakshmi Subramanian, Chief Scientific Officer, Swasti Veda Private Limited, Mumbai, Dr. G. Sridevi, Associate Professor, Department of Plant Biotechnology, School of Biotechnology, Madurai Kamaraj University, Madurai, and also Dr. D. Victor Arokyadas, HOD Department of Biochemistry, aided, and Dr. S. Mohandas, Associate Professor and Head Self-Finance.
It's my pleasure to invite Dr. S. Mohandas, sir, Associate Professor and Head, Department of Biochemistry, PSGCAS, to address the gathering and share their insights on today's topic.
A very good morning, one and all gathered here. Sorry, good evening, one and all gathered here. Most respected dignitaries on the dais, participants, and professorial staff members. It's indeed my honor to stand before you to propose the welcome address for this valediction address. On the outset, I would like to thank our management for giving us a wonderful opportunity to organize this national seminar in a grand manner. It's my honor and pleasure to thank and welcome our secretary in absentia, who has given his inputs and his blessings for the successful conduct of the conference. It is without the help and the expertise and the knowledge of our principal in charge, Dr. Sengatuvan sir, where he guided us, motivated us, and led us the path for each and every step for this success of this conference. On behalf of everyone gathered here, I take it as an opportunity to welcome you, sir. Welcome you, sir. The resource person of today's conference, Dr. Kinney from Singapore. Even though I was not able to fully be a part of the conference for today's conference and your talk, sir, I do appreciate your expertise in the inaugural address and your motivation for the participating students. And I'm sure that the students will really appreciate and really follow the paths of your words. Welcome you, sir. To talk about Dr. Karnagaran, I have no words. I'm the student of Dr. Karnagaran, and I am on this outset gathering, I would like to say that I am the last student of his MSc project. When I was doing my MSc project, sir was his guide, and the time that he was also completed his PhD, I think if I could remember it correct, it was from Krishna Devaraya University. Whenever we come back with a report, sir usually takes his PhD thesis and he usually shows his PhD thesis as a guideline. You should do like this, you should draw the graph like this. And we usually go to Vadavalli for doing the graph. At that time, near TNAU, we had only one center which was doing the graph, that was in Graph Prism. So that was the motivation, or that was the seed that he imbibed me within us for our project. Even morning when he came, the first thing he asked was, what was the topic that gave it to you? And I was able to recall it was the effect of organic acids on the germination pattern of horse gram and Bengal gram. And that was the idea. He imbibed us with three of our friends, myself and one more Began, Dr. Began, and Mr. Anbu Jeevakumar. So thank you for us to coming back to this institution, sir. And really, thank you for bringing back the memories of from us. Thank you very much, sir. And Dr. Lakshmi Narayanasamy, sir, from Dalmia Center. It was wonderful to have, to hear you, sir, and the process of creating the plant medicines and the formulating of the plant medicines for naturopathy and for the treatment of psoriasis and patenting the formulations, it's indeed a great work. And we would like to coordinate with you in future so that we, the Department of Biochemistry, most of the faculties are working on the medicinal plants. Even though our concept of idea was the work of cancer-related topic and diabetes, we would like to explore your expertise for the action of psoriasis. And as you said that during the discussion, you said that I do not know the molecular mechanism of the action of the plant extract. We would like to coordinate with you, and we would like to work on that aspect so that we can evaluate and we can divulge into the process of mechanism of action, sir. We are looking out for it. Thank you very much, sir. And Dr. Sridevi, I have spoken to her very long time in phone, daily, usually during the lockdown period. We and myself and Dr. Sridevi was working on a project where we applied for nutraceutical project in TNSST. We usually talk over phone for daily half an hour for months together. But it's the first time I'm able to communicate with you in person. Welcome you to the alma mater, and thank you, the participants, from coming all the way from various institutions. Thank you for everyone, and I take this opportunity to welcome you everyone, each and everyone, those who have participated, those who have presented your papers in this national conference. I'm looking out forward for more meeting with you in the future. Welcome you all for this valediction function. Welcome you all. Thank you.
With great respect, I would like to invite Professor Devarajan Karunagaran sir to present the valedictory address. We look forward to your concluding words for today's gathering, sir.
I was wondering, already I gave a lecture. Do you want to hear me again? Then Victor told me, sir, say a few words to students and things like that. Anyway, it is really on a personal note, homecoming for me, and I think Dr. Sridevi as well, though under different these things. She was a student here, I was a faculty here, but both of us came back to meet some of our old friends and students. It is always a pleasure to be among students. That has been my passion as well. That's why I always tell the students to pursue their passion. Being in IIT, you know, a lot of people come to us for some kind of counseling, apart from our regular teaching work, administrative or whatever work that we do. We always tell them to pursue their passion. It doesn't matter. Sometimes even if I can share some kind of experience, even long back, when I was doing MSc in JIPMER Pondicherry, I was doing MSc Biochemistry. Already I was regretting because it is a 3-year course. Normally MSc means 2-year course. So it was a 3-year course, and some of us were kind of regretting. And one of my seniors, he did MSc Biochemistry and came out with first class with distinction. And then when he went out, he joined Andhra Bank as a manager. So everybody was kind of ridiculing that kind of a decision. interestingly, I met him after many, many years. Then I asked him this question, hey, you did so well in biochemistry, and then all of a sudden, like a crazy fellow, you went to a bank. What is the use? Why did you do like this? Do you really regret for that? He told me, no. He said he worked in the bank on some division where he had to make decisions on giving loans to some insurance companies, some diseases, this, that, even on the agriculture sector and all that. In all these places, his knowledge actually helped, it seems, and he cherished. So what I am trying to say is, there are many examples. You take people, there are very famous celebrities also who have studied something but are pursuing something else, and they are happy. Why not? Not that I want everybody to deviate from your subject and go. It's not like that. If you are interested in that, like especially I was impressed with what Dr. Sridevi also told, when we come and give a talk, if it inspires even 10% of the students to become researchers or something, the purpose is served. So if you have that spark and if you have the interest, certainly you need to pursue. If you don't have, don't think that it is a waste. That's not true. Whatever you learn is useful. With the IIT example, if you want me to tell, all the IITs, the older IITs especially, they have decided on one thing. You might have come to pursue civil engineering, mechanical engineering, or electrical engineering, or biomedical engineering, or whatever. There are so many branches in engineering colleges. But all the students should have one subject on life sciences. Students were obviously very unhappy. They don't want to take, because I want to be a civil engineer or mechanical engineer. Why should I study life science? This is their thought. But why these directors and all the learned people decided like this? Even I was wondering as a teacher. So when this course was introduced, I volunteered to do that. And we all sat together and tried to find out why this kind of a decision was taken. So when we had a discussion, we came to know that even in what is the best university in the world, people will say Harvard. You go to their mechanical engineering department in the Harvard University, more than 60% of the faculty in that department are pursuing some work related to biology, it looks like. Because you see, ultimately, whatever you study, at one point of time, you may want to apply something to the human welfare. At that time, if these words are totally new to you, you will have total inhibition to pursue that. Your passion may change with the age. It is possible. So nothing is a waste. Don't consider that as a waste. At a later time point, if your passion changes or if you want to pursue, you should not be thinking that this is a waste. No. Education is, even in Harvard, again, to quote that, education is not about simply gathering some bookish knowledge and all that. The major aim, as stated, written statement in Harvard also, is character building. So, that is more important. So, whatever you learn, you keep that in mind, and if there is a spark to pursue research, yes, do it. There you should not be thinking about only money. If that is the motive, then you will not find any pleasure in that. It is not that I am not saying that one should not go behind money. Money is important, obviously. But that should not be the only goal. What is your passion, you pursue. What you think as, like I started the this talk about how students are persuaded. So we always tell them, even to the parents we used to tell that allow the children to pursue what they want. Of course, these days it is different. These days, even at the +2 level, the students are more or less very good compared to our generation. We had to depend upon advice from others. These days, you have better exposure because we were not born with television or computer or something like that, but you are. So you are able to kind of decide. So once you decide like that, you can pursue on that. That will give you more happiness. This is the lesson I want to say. And these kind of conference and other things give an opportunity to interact with people and to gain more experience on various perspectives of the same problem. Somebody also in the lectures told that when you, I think Dr. Kinney mentioned about it, when you read something from different people, you get different perspectives. As a student, even in those days, I used to read any topic. I used to read 3 books or 4 books because I wanted to see the same topic, how it is discussed by different people. Then I form my own this thing. Even today, I am doing that. If I want to learn about something, as I mentioned, we are always learners. So when I want to learn something, I want to look at different perspectives and see which one suits. In this manner, you will try to learn more and more. So this is what is a piece of suggestion that I may be able to give, and I hope you all enjoyed, and I hope you do not feel that coming here and sitting down is a waste of time, but indeed it was useful. If you feel that, the whole thing can be considered successful. I hope it was like that. For us also, it was successful. Like I mentioned, it was a big homecoming. We should cherish with some of the old friends, coming back with all those memories. And also, if we have done some useful purpose of encouraging you into something, then it is done. Thank you.
Thank you, sir, for sharing your sweet memories and enlightening words. It's time for feedback. Participants from other institutions can share your views on today's conference.
Good evening, everyone. I am Sajita and I'm pursuing my Masters in Biochemistry in Bharat University, Coimbatore. So this one-day national conference on the topic Innovations and Emerging Trends in Natural Therapeutics and Molecular Biology and Structural Analysis of Biomolecules organized by the Department of Biochemistry was a wonderful conference. So during this conference, we got to learn about a lot of topics, especially microRNA. So to be honest, even just from this year, even our topics are getting updated with microRNA. So rather than just theory, today in this session we got to know a lot about microRNA. And apart from it, the drug formulations are from herbal plant-based drug formulations and oncology and toxins and venoms, how they are being used from in the field of therapeutics. All these have actually ignited our minds in terms of now we are getting a lot of ideas for projects and research oriented ideas. So this is actually a very great opportunity for all of us, and it is a privilege to attend this conference. I studied my UG over here, and I know how hard our staffs and our students work behind bringing together this one-day national conference. And this has been a great event, I thank all, I thank our staffs and I thank our principal in charge, our secretary, the head of the department of both biochemistry, SF and aided, and the staffs and my friends for bringing together this event. And this was a very great event, and I hope it ignited our minds for in the stream of life science and in other aspects also. Thank you.
Good evening all, everyone present here and experts also. It's me, my name is Sai Sri Krishna from Sri Sakti Institute of Engineering Technology. I'm pursuing third year BTech Biotechnology, and I'm truly grateful to attend the conference, national conference on natural therapeutics and molecular biological and structure of biomolecules. And it's truly about innovative ideas, and the experts gives the motivated speech to all the students to research about the new field of life science. And I thanks the PSGCAS and Department of Biochemistry to giving this opportunity for a wonderful conference. Thank you and all.
Now it's the time to recognize the prize winners. I would like to welcome Professor Devarajan Karunagaran sir to acknowledge the prize winners of poster presentation. First prize goes to Arul Prakash, Research Scholar, Department of Biochemistry, PSGCAS. The second prize goes to S. Sai Krishnan, Department of Biotechnology, Sri Shakti Institute of Engineering and Technology. I would like to welcome our Principal in charge, Sir, Mr. Sengutwan, to honor the oral presentation prize winners. First prize goes to Karthikeyan M, Department of Biochemistry, PSGCAS. Second prize goes to Priyadashni K and Vidyashri S, Department of Microbiology, JSS Academy of Higher Education and Research, Ooty. Third prize goes to Aishwarya G. Nambiar, Research Scholar, PSGCAS. Thank you, sir. Thank you, ma'am. The prize winners of oral presentation online mode. First prize goes to Abira R, Department of Biotechnology, Vellore Institute of Technology. Second prize goes to Dharani Ambigai, Researcher, PSGCAS. Third prize goes to Vidyashri M, Department of Biochemistry, NGP Arts and Science College. The e-certificates will be sent to the winners through email. Thank you.
Gratitude is the best attitude. I now request Dr. R. Sivaprasad sir, Associate Professor, Department of Biochemistry, PSGCAS, to take the stage and provide the vote of thanks.
Good evening. I deem it a great honor and privilege to propose a vote of thanks at the culmination of this memorable event. I take this opportunity to thank our management, Secretary Dr. T. Kannayan, Principal in charge Dr. Sengutwan, HOD Dr. Victor Arogidas, for creating a platform like IETNMS to share and spread the varied insights and experiences pertinent to the theme of the event. I also take this opportunity to thank the staff members, students of various departments inside and outside for participating and making the event a grand success. I also take the opportunity to thank our resource persons, Professor Kenny, Professor Karunagaran, Professor Subramaniam, and Professor Sridevi for their valuable inputs and sharing their expertise in their respective area of research. I also take the opportunity to thank the organizing committee staff members and student volunteers and the maintenance department for their overwhelming help and support rendered for the grand success of this event. Thank you one and all.
Thank you, sir. Our heartfelt thanks for all participants. Dear participants, kindly collect your certificate from the registration desk. Thank you. All registered participants, kindly have your refreshment. It's time for national anthem. Everybody stands.
Participants who didn't get the registration kit, kindly